Hello everyone, welcome to this presentation about the genetic basis of conscientiousness. So let's first define what conscientiousness is. You may have heard of the term consciousness, which is spelled similarly, but it's a different thing than conscientiousness. So consciousness, which is not what we're talking about, is the just in general like being aware of your own existence. But conscientiousness, which is what we're talking about here, is a personality trait that is generally defined as being responsible, diligent, hardworking. So if you're high on conscientiousness, you're more likely to want to do a good job on the tasks that you are assigned, and you want to take your obligations seriously. Now, each field of psychology has defined conscientiousness as a more specific trait. So, for example, developmental psychologists have used the concept of effortful control. So instead of saying conscientiousness, they define it a little differently in their surveys, and they call it effortful control. And then social psychologists have used self-control, and positive psychology more recently has called it grit. And personality psychologists have just called it conscientiousness in general. So the first question that we want to ask is if each definition of conscientiousness is a separate trait or tendency, or if all of these characteristics are all correlated and they reflect a common tendency or trait. So that's what this study attempted to do. This was a study of 2,000 Japanese twin pairs. So what they did here was they took people and they did surveys to measure their conscientiousness, and then they correlated that to their effortful control, to their self-control and grit as defined by each field of psychology. So for example, here we can see conscientiousness is 1, and then it is being correlated to 2, and 2 is effortful control. So conscientiousness and effortful control have correlation of 0.78, right? So that's pretty high. And for all of the other ones being correlated to each other, you can see it's fairly high. It's from about 0.7 to 0.8. So it suggests that conscientiousness, as defined by all these fields, is a general tendency or a common trait. So then this is there was a twin study. So what they did was they had monozygotic twins, which are identical twins, so they're genetically identical, and then dizygotic twins, which are fraternal twins, so they were not born together, but they only share half of their own variable DNA. So what you can see here is that this number here is the concordance, meaning the conscientiousness, how correlated is it between siblings. So you can see for fraternal twins, the correlation on conscientiousness, effortful control, self-control, grit, it's around 0.3 to 0.35. And then when you look at twins that are identical in terms of their genetics, so identical twins, it jumps to around 0.55 to 0.6. So there's almost not quite, but almost a doubling of the concordance when we go from fraternal twins to identical twins, so which shows that there's probably a strong genetic component that is leading to the variation among people with this trait. And then that same study here asked the question of how much of the variation is genetic versus environmental. And for different definitions of conscientiousness, it differs a little bit, but generally it's 60 to 65% genetic and 40 to 35% environments. Another similar study, this one, they had people, twins that grew up apart, so that's the A's, and twins that grew up together, so that's the T's. So if we examine only the twins that grew up together, so the T's, and we look at the fraternal ones and compare them to the identical ones, we can see that the fraternal ones had a concordance in terms of conscientiousness of 0.11, and then it jumped up to 0.47 with identical twins. So shows a pretty strong effect from genetics. But we also know that environment plays a role here, because if you only look at the monozygotic, so only the identical twins, and you look at the ones that grew up together versus apart, the ones that grew up together had a concordance of 0.47. The ones that grew up apart had a concordance of 0.19. So environment also seems to have an effect, because if your genetics are the same but your environment's different, the concordance is not as high. Now, just as a, we're talking about conscientiousness, but they also did openness and agreeableness. For openness, it's very interesting. The identical twins that grew up together have a correlation of 0.51, and the ones that grew up apart still have a pretty high correlation of 0.43. It shows that basically for openness, it's a trait that doesn't have as much environmental contribution as conscientiousness. The study asked the same question of how heritable is it. For males, they estimated to be 41. For females, 11%. For younger than 60 years old, it's 32, and then older than 60 years old, it's 13%. So that's how much genetics is going to contribute to the variability of the traits. So next one here talks about similar sort of thing, but they break down conscientiousness into specific components or behaviors that are not the same as we previously discussed. So they measure specifically competence, which is sort of what knowing what you're doing, and then order, dutifulness, achievement, striving, self-discipline, and deliberation. You can see that the correlations for the identical twins are much higher than the fraternal twins. So 0.08 versus 0.38, 0.06 versus 0.16, 0.13 as opposed to 0.40. They're all relatively higher in the identical group. What's striking to me is the self-discipline part. Fraternal twins for self-discipline have a correlation of 0.03, and it jumps to 0.53 if they are identical. So that shows that the self-discipline component of conscientiousness is probably the most heritable one, as defined by these components here. So now let's talk about some genome-wide association studies. There was a meta-analysis of many genome-wide association studies, and they identified this gene, a single nucleotide polymorphism in this gene called catenol2. It's a gene that codes for a protein that's involved in development of neurons, migration of neurons, axonal growth, dendritic pruning. So it's basically important for the development of the nervous system, which would be development of the brain and perhaps circuits related to self-discipline and conscientiousness. There's another study, so that was one study. Another one just did one genome association study, and they found 3 single nucleotide polymorphisms. There was one on a gene called insulin-like growth factor 2 mRNA binding protein 3. So it's not insulin-like growth factor itself, but it's a protein that binds to the mRNA, and it's involved in its function and its expression. It's not clear why this gene was found significant results, but they tested this on another cohort in the same study that was not replicated. Another gene, another single nucleotide polymorphism. This one I could not find any information about the gene or the SNP, but they replicated it in another cohort, but then it was just opposite effects, so it's kind of awkward. There's another single nucleotide polymorphism on this gene. It codes for a protein that's important for the initiation of transcription, so it will help basically RNA polymerase II to initiate transcription. And in this study, was, by the way, done on Korean women only. Okay, and they did not find a link with catnol 2, as was previously found in the previous studies. So as you can see here, the common theme of a lot of this is that you just have results that are not replicated. So it's a little bit, it can be a little bit disappointing at times. One more genome-wide association study found these two SNPs. One of them is on this protein. That's the gene is called SMOC1. I don't remember what it's called. I believe it's, let's see what it's called. Secreted modular calcium binding protein 1. So basically is a protein that binds to calcium, and it's going to be involved in formation of bones, as you can imagine, outside the nervous system. But it's been shown that when neurons secrete this protein, it's going to regulate angiogenesis and neurovascular development. What that means is the formation of blood vessels that are going to supply the nervous system. So these blood vessels would form, that process of blood vessels forming to supply the nervous system would in part be regulated by this protein. And then this last SNP is on a gene called DYRK1A. It's a gene that is implicated in a rare form of intellectual disability, and also there's been some correlations with increased risk of Alzheimer's. So that's it for the genome-wide association studies, and here are so this is the one that found catnol 2. This is the Korean women study that found the 3 SNPs. And this is the last one with the calcium binding protein and DYRK1A. So now candidate gene studies. The first candidate gene study examined the dopamine 4 receptor gene. So dopamine, the neuromodulator, has 5 types of receptors. It goes from D1, D2, all the way to D5. The D4 receptor is, in animals, it's been shown to be expressed in areas of the brain that are involved in exploratory behavior. Okay. In humans, it's generally believed to be involved in areas that regulate cognition and emotion. Now, this gene for the 4th dopamine receptor, it has an exon, exon 3. Exon is the portion of the gene that is going to be expressed. You can see that they call it here 48 base pair variable number tandem repeat. What that means is that there's a 48 base pair long sequence of nucleotides, and it's going to be repeated in this exon. So some people might have it repeated twice. Some people might have that 48 base pair sequence have repeated 3 times, 4, 5, up to 11. Okay, so that's why it's called a variable number tandem repeat, because it tends to vary among people. So it's been demonstrated that people who have higher number of repeats on this exon are going to be higher on novelty-seeking behavior. You can imagine that novelty-seeking behavior probably has something to do with exploratory behavior in animals. So that might be the version of it that you see in humans. So the study, what they did was they said, okay, if someone has 2 to 5 repeats, we're going to call that S allele, and if someone has 6 to 8 repeats, we're going to call that the L allele for large number of repeats. And so since people have 2 copies of this gene, one from each parent, they could either be SS, LL, or SL, based on how the researchers have defined this as. The question that they were asking was really about novelty seeking. However, it's been shown that conscientiousness is negatively correlated with novelty seeking. What that means is that if you generally are more thrill-seeking person, you're probably not as disciplined. That's been the correlation found, and that people who are more novelty-seeking are more extroverted. And then what they found was that people with S allele were more conscientious, and people with the L allele, so more repeats, were more extroverted, and less repeats, more conscientious. Here is a novelty-seeking score. So if they're further to the right, they're more novelty-seeking than if they're to the left. And this is kind of a distribution, like a bell curve almost, right? And you can see that this is the L allele here, meaning more repeats. So if someone has more repeats, distribution is going to be, for novelty seeking, is going to be like this. If you have less repeats, so if you have 2, 3, 4, or 5, it's going to be kind of to the left. So you can see the distribution is going to be displaced whenever you have a larger allele or more repeats. So it's not that every single person with more repeats is necessarily always going to be more novelty seeking or less conscientious than people with less repeats. It's just that in the distribution, it's a little bit displaced toward the side of being more novelty seeking. So the next gene that we will talk about is another study. This is the serotonin 3A receptor. So this is a ligand-gated ion channel. So serotonin is the 5-HT is the ligand. It comes and binds to the receptor, and then the receptor allows either sodium or calcium in, or potentially potassium to leave. Now they examined this with regards to e-cigarette usage. So their groups were controls, meaning people who have never used e-cigarettes, and then people who have used e-cigarettes, or probably currently do. And it's interesting because e-cigarettes, the substance that's in there is nicotine, and nicotine happens to actually be an antagonist of this serotonin 3A receptor. So it binds to it like serotonin does, but it blocks it. It doesn't allow serotonin to bind, and it also doesn't let ions to flow through. They were concerned with two polymorphisms, two single nucleotide polymorphism within this gene. These are the two, in case you want to look them up, but it's just numbers. There's not really no name for it. But for the first polymorphism, this is what they found. So the red is the controls, the blue is the e-cigarette users, and then this y-axis is conscientiousness scale. And these letters at the bottom, so A means that at that spot where there's variability, that SNP spot, there's an adenine. Right? And then this means that there's a thymine. So on one of their chromosomes, it was an adenine, and then on the chromosome that's homologous they got from the parent is thymine. And then you can see here that this is if they have both of them have adenine and both of them have thymine. So for the heterozygous groups, you can see that the people who were not e-cigarette users were generally more conscientious than the cigarette users, which is expected, right? I mean, in general, that is a finding you'd expect, because people with e-cigarette usage, it generally would show that they have less self-discipline and therefore less conscientiousness. And that trend is true with all of these except people who have both thymine. For people who have both thymine, there's no statistically significant difference in their conscientiousness, whether there's e-cigarette users or not. And then what's interesting is that you might see the graph and say, okay, look, the people who have adenine homozygous are probably more conscientious than the control people who have other, at least one thymine. But statistically, this result is not significant. So it's not just the mean that matters; it's also the sample size and the distribution. And so these three are actually similar to each other in terms of statistical significance. But however, this result here, where you can see that the e-cigarette users that have both adenine are actually less conscientious than the ones that have at least one thymine. So this result here, this difference is significant, but this is not significant. So it's kind of a puzzling effect. This is the other single nucleotide polymorphism within the same gene. again, serotonin receptor. At least one of the serotonin receptors. With this one, everyone, not everyone, but on average, the people who are not cigarette smokers are going to be more conscientious than the people who are. And so that's true with all these, all the genotypes. And you can see that the people who have cytosine thiamine in our control group are going to have basically higher conscientiousness than everyone except the thiamine control group. And it appears that the people who have cytosine for e-cigarette users have higher conscientiousness than the people who have at least one thiamine, who are also cigarette users. So there's not much to be gained from this in terms of a full story on how this works, because we don't quite know what those single nucleotide polymorphisms, how they would change the shape of the receptor, perhaps, because if you're changing one nucleotide sequence, you might code for a different amino acid. It might change the shape a little bit, might change the neurotransmission of serotonin a little bit, might change the sensitivity of that receptor, might lead to a difference in behavior, but we don't have a full story on that. We just have correlations like this. The last study that is a candidate gene study is examining endocannabinoid receptors. So it's specifically looking at CNR1 gene, and that gene makes a protein. The protein is this one. It's CB1, cannabinoid receptor 1. So here's how that works. A presynaptic neuron will be sending either GABA or glutamate to a postsynaptic neuron. And the postsynaptic neuron will receive the GABA or glutamate, and once it's received it, it's going to send back endocannabinoids. And those endocannabinoids are going to bind to these endocannabinoid receptors, and then the endocannabinoids are going to cause the presynaptic neuron to stop sending the glutamate or GABA. So you can imagine it as the presynaptic neuron says, hey, here's some GABA or glutamate, and the postsynaptic neuron sends endocannabinoids saying, okay, stop sending the GABA or glutamate, I received it. Essentially kind of a conversation in terms of an analogy. Now, you can imagine that if there's a single nucleotide polymorphism within this endocannabinoid receptor gene, that it would cause, perhaps a difference in the expression levels, maybe. If it's, in a coding region, it would cause maybe a slight difference in the shape of the receptor, and might impact its function. But the SNPs we're going to look at are actually not on the coding regions. So meaning that it's probably changing the expression levels of it in some way. And so this gene has been implicated in disorders that are generally going to involve high neuroticism and anxiety, those types of psychiatric disorders. And you can kind of guess that would be expected, because these endocannabinoid receptors are important in the modulation of activity within GABA and glutamate synapses. And GABA and glutamate are the major, for GABA, inhibitory, and glutamate excitatory neurochemicals in the central nervous system. So if those synapses do not have sufficient amount, or have too much endocannabinoid receptors, it could dysregulate that system, and you could have too much excitation, and that would lead to anxiety disorders. So here they have a nice diagram of showing the entire gene, and they have these single nucleotide polymorphisms along the gene. These are all the SNPs that you can find there, and they basically correlated these single nucleotide polymorphisms along this gene with people's conscientiousness. And this was only among African Americans, interestingly. They only found correlations between these first three single nucleotide polymorphisms. So this first one is in the 5' kind of in the promoter region. Probably if you had a single nucleotide polymorphism here, you might impact the expression levels of it into mRNA. And then there was in the upstream region, there were 2 SNPs that were also correlated. But again, those are on non-coding regions. We don't know how they impact expression. We don't know much about that. So here are the references for the candidate gene studies. This is the dopamine 4 receptor study. This is the e-cigarettes, the serotonin 1 study, and then this is the endocannabinoid study. In general, I found it very difficult to find good sources, and not good sources, but in general, there were just not a lot of studies that were asking the question, what is the genetic basis of conscientiousness? If you notice the studies that I mentioned, almost none of them have a central focus on conscientiousness. For example, this one here, they were looking for personality in general, and conscientiousness just happened to be part of that. And these guys were looking at e-cigarette usage and personality. And these guys were looking at novelty seeking. And the genome-wide association studies were generally just about personality in general. So I think that part of the problem in this literature is that no one's really dug deep into conscientiousness specifically, and no one's really asked the question well enough. So I think you can't really get a good answer if you're not asking a very specific question. And I think, conscientiousness is an interesting trait because it's generally something that is positive and it's adaptive. If you have a stressor in your environment, if you actually deal with it and try to get the work done, it's a lot better than avoiding it. That's to an extent that's true. You don't want to go over the extreme with conscientiousness. So it's an interesting characteristic. It's an important one, and I think it's one that we don't know enough about the genetic basis of it. And if we design studies that are more focused on it specifically, I think we will find interesting associations. I just think no one's really went down that road long enough or rigorously enough. But that's basically the best of what I could gather on the literature surrounding it. So I hope you learned something and enjoyed it. Thank you for watching.